Welcome to Inside Startup Investing, the only podcast where you can invest in every guest. On this episode, I will be speaking with the founder and CEO of Siren Biotechnology, Dr. Nicole Paulk. I've talked to almost 1,000 startups in the past decade, but Siren Biotechnology, I personally think, may be one of the most exciting companies that has the potential to create pretty exceptional monetary and human impact in the next decade. And I am delighted for Siren to be one of the latest Kingscrowd Capital portfolio companies. Siren is an advanced biotech company backed by some of the biggest Silicon Valley investors that has discovered a potential mechanism to tackle brain cancer and other cancers with an extremely targeted therapy that could someday actually be a cure. Now, they are not there yet. All studies have only been in animals yet, but as you will hear in this episode, the data and signals are extremely exciting for cancer patients and doctors alike. A few things I really want you to pay attention to in this episode are first, listen to how Dr. Paulk and her team have identified safe human viruses that they can leverage to help the body tackle cancer in an almost organic, natural way by helping to provide the body with directions on how to attack and kill cancer cells. Both virus-based therapies and gene-based therapies were a thing of science fiction just a decade ago, but they are starting to become real therapies that the FDA is clearing and are helping patients today. Perhaps just as impactful as this therapy, having potential curing abilities in the future, what is also so exciting about this drug is that it is a deeply targeted therapy. As many who have had the unfortunate reality of facing cancer or having a friend or family member experience it know, treatment with things like chemo has acutely harmful effects across the entire body, hurting more than just the cancer cells. A targeted therapy with far less side effects in and of itself could be a major win for cancer patients. Lastly, just to use one proxy, Keytruda is a leading cancer drug today in the immunotherapy space and currently produces nearly $30 billion a year in sales. This drug that Siren is developing may actually be an even better, more advanced drug in the future for cancer treatment. Put simply, the upside, if Siren can do what it has showed early signs of being capable of, could be massive. There is still a ton of risk in investing in an early stage biotech like this, but it is hard to deny the upside potential of such an impactful new cancer therapy. So with that, let's get on to the show and welcome Dr. Paulk. Dr. Nicole Paulk, thanks so much for joining us today.
Yeah, happy to be here.
So for those who don't know, tell us a little bit about your incredible business and how you came to found it.
Yeah, so if folks don't know, prior to this, I was actually a professor of AAV gene therapy at UCSF. Had no grand visions of starting a company and being a CEO. That wasn't on my bingo card. But we started Siren actually in the peak of COVID, right? When you were still Lysoling off your groceries in month 2, right? The very beginning when we didn't know what was going on. There was no vaccine yet and everything was still kind of panicky. And it was a project in my lab that we had already been working on and that our clinical colleagues are really excited about. And it was really, we didn't know it at the time necessarily, but it was to kind of do two of the world's firsts. We wanted to make the world's first AAV in oncology, right? So using an AAV gene therapy, these are viruses that we can use to deliver genetic medicines, but they're viruses that don't make you sick, which is why we use them in a medical case. Using viruses to deliver genetic medicines, but now in cancer for the first time. This virus has only ever been used to treat genetic disorders in little kids, but it had never been used in a cancer setting. And we also wanted to make the world's first AAV that could be used to treat more than one thing. So all the other AAV gene therapies on Earth, whether they're happening in academia or in industry or in government consortia, they're all one for one, right? One virus can only be used to treat one disease. And we were like, well, what if we made one that could treat hundreds or thousands of different indications, all from the exact same single individual virus? Then you could kind of produce this stuff at scale, and now all of the economies of scale and metrics would matter for the first time. And so wanted to kind of do those two things. And again, all of this was still from within academia, and then COVID hit, and it was I don't even know, month 2, 3, 4, 5, 6, and we were just crawling out of our skin, wanting to get back to work, but we weren't considered essential workers, despite the fact that I was a professor of virology, of viruses, right? And our clinical colleagues were just gosh, some of the data that you're getting, we were working in brain cancer at the time, still are. Our clinical colleagues were this is really good data. We should really consider running a clinical trial, but like please go start a startup and go get like real money so that we can move much faster, right? Clinical trials in academia are often quite slow. And they were like, let's go get real money and put jet fuel on this because this is looking really promising. And so we decided to spin out of academia back in early 2020 and have been off to the races ever since. So, and I decided to resign.
Let's break some of this down. So first off, you say there are viruses that could go into our bodies and not make us sick. What are these viruses?
Right? That sounds weird, right? Particularly given like, the cultural way we interact with viruses, right? When we hear about them in movies and in magazines and in songs and in books and all of our like cultural references, it's viruses are always sick, right? That's the party line. Viruses are always bad and they always make you sick. In reality, most of that is there's like 30 to 50 viruses that are that, right? That are gnarly and are nasty and are scary, and measles, mumps, and polio, and smallpox, and chickenpox, and all of the cold virus, and all these viruses, COVID, right? All these viruses that get a lot of airtime, but that's not the vast majority of viruses on Earth. Most of the viruses that have ever been discovered do not make humans sick. They're still good at getting inside of you, but they don't make you sick, right? You're crawling in them. They're everywhere. Right? So they're very good at getting inside of you, and they're very engineerable. They're very, very simple things. So it's very easy to, kind of it's very similar to like coding on a computer with like zeros and ones, right? It's very easy to engineer and program and code these things, only instead of zeros and ones, right? It's A's, T's, G's, and C's, right? The genetic code in your genome. And so we can use these viruses. We use a very specific one called AAV. The nuance of what that stands for and what it means doesn't matter. But it's a very special virus that doesn't cause disease in humans. Also doesn't cause diseases in animals, doesn't cause diseases in plants. Like it just doesn't cause disease in anything. But it's still very good at getting inside of us. And so we can take it, use it as like a little programmable FedEx truck, and we can tell it like, I would like you to go only to the lung and the heart, but nowhere else. Or I want you to go to the kidney and the ovaries. Or I would like you to go to the brain and actually every single other organ in the body. I want you to go everywhere. Like you can program these viruses to go where you'd like and not where you wouldn't. And you can have them deliver basically little packages, little payloads of whatever you'd like. In this case, we can deliver an anti-cancer drug. And we can have them do very, very targeted delivery in your body. And this is very different from how people normally think of cancer drugs, about like chemo, where it's like, oh man, I'm gonna get this like IV infusion into my arm and it's gonna make my whole body sick, because it will, right? We deliver these systemically throughout your whole body, and they're not targeted, they're not specific for anything. And so they make all the cells in your body, not just your cancer cells, they make even your healthy cells very sick. And so that's why you feel like trash after you get chemo, but you can use a virus to do very, very targeted delivery in a really engineerable way. So it's a whole field of viral gene therapy that I think most folks maybe haven't heard of before, but it's starting to become a thing. There are now 5 FDA-approved viruses where if you had that disease, you could go to your doctor and get a prescription for a virus to treat your disease.
In terms of how you discovered this virus and its potential ability to be able to go a lot of different places and to be programmed, so on and so forth, is this a virus that is already being used in these other disease sets? Or have you discovered this one very specific one? And what about it makes it so unique that allows you to do this very specific delivery?
Yeah. So we did not discover this class of viruses. So AAVs, adeno-associated viruses. It's this family of viruses and called dependoviruses and parvoviruses. We did not discover them. They've already been used in the clinic. They've been used to treat like 10,000 patients across 200 clinical trials, just never in a cancer trial before. They've only ever been used to treat, for the most part, rare genetic disorders in children. But they've been used in adults, they've been used in children, they've been used for local delivery and systemic, so intravenous delivery. They've been used in like late stage and very acute diseases. They've been used to treat just about every organ you can imagine in the body, but again, only for single gene genetic disorders and never in an oncology setting for a cancer. And so we're the first company to kind of bring this whole new modality into the oncology setting and start thinking about treating cancer patients for the first time with this virus.
Now, the other part of this, so we talk about virus, and then there's the gene part of it that you mentioned. I know that there's a few kind of gene editing products out there, and there's been some controversy about their effectiveness and different things like that. Can you talk about kind of the gene therapy class and how you think about it and how effective you think it may be? Yeah,
so if we take like the mothership concept of gene therapy, there's like different subcategories of it. There's like using viruses to deliver genetic medicines. You can actually use cells. So folks may have heard of things like cell therapy, immunotherapy. That's a type of gene therapy. There's also a special sub-branch of gene therapy called gene editing gene therapy, where we actually edit your genes inside your body. Where we can go in and we can use, and it doesn't have to be viruses that deliver the gene editors, right? The actual tools that do the cutting up of your genome. It can be non-viral as well. But there's like all these different flavors. There's like viral and non-viral. There's cell-based, where you use a cell to deliver it. And then within gene therapy, you can deliver a functional copy of a gene that maybe a child was born with. But you don't actually edit their genome. You just give them an extra copy. Like they were missing a copy, maybe like mom didn't have that gene in her particular genome or something, and when she passed it on to the baby just didn't get that gene, and so they're missing it, but they needed it, and so now they're sick. You can do gene transfer gene therapy, where you deliver them a functional copy of that gene they're missing. It doesn't integrate into that baby's genome, so it's not like they can then pass it on. They're just giving you an extra copy that like kind of lives in the cell but doesn't actually integrate into the chromosomes. Or you can do gene editing gene therapy, where you actually edit their genome and you plop it in to their DNA. So now it is something that will be in their DNA, but it still wouldn't be heritable, because in order for it to be heritable, right, it has to happen in either a sperm or an egg. So if you do that in like a skin cell or a liver cell or a brain cell, you still wouldn't inherit it. You would benefit from it as a patient, but you wouldn't be able to then, as that patient, pass that on to your children because we didn't actually edit sperm and eggs. So we're still not, as a society yet, maybe one day we will be, but we're still not yet as a society doing germline gene editing, where you could actually inherit it. We're just editing what's called your somatic cells in your body after you're born to kind of correct genetic disorders. And so the class of gene therapy that Siren has is a gene transfer gene therapy, not a gene edit gene therapy. So we're not touching your genome. Your genome's going to stay your genome. It is what it is. We're actually just delivering an already kind of well-established and well-known anti-cancer drug called a cytokine. This is something your body already makes to fight tumors. So every day, like even you, Chris, right? Don't freak out. You have a tumor right now. You have cancer, but it's okay, right? It's a single cell. You have them all over your body, and you've had them every day of your life. So is everyone who's listening to this. You've had a tumor, you've got a tumor right now. It's okay. They're single cells. Your immune system is really good at actually sniffing out and finding those individual cells, because those individual cells have like little flags on them that say like, I'm not self, I'm a tumor, something's up with me. And your immune system can come by, swim, find it, detect that, and kind of like a lock and key mechanism be like, something's up with that guy. I'm going to chew it up while it's still an individual tumor cell before it has a chance to become like this large tumor mass that's like the size of a baseball and you have symptoms. Right? So your immune system is very good at doing this already. And the way it does this, the way it recognizes those cells, those individual tumor cells, are by little cytokines that are secreted by the healthy cells that surround that tumor cell. They secrete these little chemical signals to the immune system, and the immune system's like, haha, I received your little package. I will come and I will chew up that tumor cell. Right? So your body already is like, mwah, chef's kiss, phenomenal at fighting cancer. It does it every single day, and it's mediated by cytokines. And so we already use these particular chemicals to fight tumors already, but they're very, very potent and they're very strong, and we want to deliver them right to the spot where we want them, right? Only at tumors and nowhere else in the body. And so we can use viruses to basically help your immune system do what it already knows how to do, which is fight cancers, but do it better. And deliver these cytokines using viruses. And the only reason you need to do this in the first place, because like how did you actually get a tumor if I just told you your body's very good at fighting cancer? What can happen is that individual tumor cell, through all kinds of like nuance and PhD level chemistry, doesn't matter, right? Through all kinds of mechanisms, it can basically evolve a little invisibility cloak that shields those little flags that are on its surface that say, I'm not self and I'm a tumor. But if it has a little invisibility cloak over it, no one can see it. And so your immune system can't float by, won't detect all of those little chemical signals, won't get the call, won't get the siren, right? The fire alarm, hence the name of our company, right? It won't sense the fire alarm, and it'll just float by, and now that single cell has time, and so now it can grow and divide and grow and divide and grow and divide and become this big tumor, and now you get symptoms, and right now you go to the hospital, and that's how you end up getting tumors. But you can retreat your immune system to basically say like, no, there's a tumor there. You can't see it because of an invisibility cloak, but there's a tumor there, and so you can deliver cytokines with a virus to that exact spot, and then your immune system will be like, haha, I can see it again, and then it will go and destroy the tumor itself. So you actually don't need things like chemo and radiation, right? You can fight tumors yourself using your own immune system, and that's like the whole concept behind immunotherapy that folks may have heard about before. Is basically re-triggering and retraining and reinvigorating your immune system to go after the tumors that are already there that it already knows how to fight.
What are the risks associated with something like this? Can things go wrong with putting a virus in your body? How do you think about that?
So any new drug always has risks. That's why we have clinical trials, right? We got phase 1 trials and then phase 2 trials, then phase 3 trials, right? So we have this whole trial schema in order to try to detect risks and minimize them, right? So phase 1 trials are in order to determine, like, is this thing safe or not? Yes, no. That's a very common misconception. Most people think phase 1 trials are actually like, we want to find a drug that's gonna cure you. Mm-mm. Phase 1 trials are to determine if the thing is safe, yes, no. Very binary, yes, no. Then if it's safe, move to phase 2 trial. Phase 2 trial is now let's find the best possible dose. And so you'll test a bunch of different doses to find, like, this is the Goldilocks. This is the sweetheart dose that works best. Then you move to a phase 3 trial, and that's the one where you're actually trying to show that, like, nope, yep, this thing is great. It works, it's efficacious, it provides benefit, and it's better than the current drug, the current standard of care, the current treatment, the current whatever it is that we normally treat that particular disease with. Now we're going to go head to head and do a showdown and see, like, is it better, right? And so all of those clinical trials are basically geared towards assessing safety, but only some of them are geared towards determining, does it work, efficacy, and those are those later stage trials. And so all of our animal studies to date show that this stuff is super, super safe. But you always have to run a phase 1 clinical trial in humans, right? A monkey's not a human, a pig's not a human, a mouse is not a human, a rat is not a human, right? They might all be mammals, right? So these are all of the types of animal models that the FDA asks you to do preclinical testing in order to assess things like safety and efficacy before you start a human clinical trial. And they are great at weeding out some things, but they will never tell you everything, right? There's nothing that is a human other than a human, right? You've got to get in humans in order to really, really find out if something is safe and efficacious. And so all of our animal studies to date look great, and we're excited to start moving towards the clinic here in 2026, and then we'll really get to see. But for, like, in broad strokes, right, these viruses have been given to, I think it's been used in over like 200 clinical trials at this point, tens of thousands of patients. But has never been tested in an oncology setting before. And so we'll kind of be the first to walk out into those, walk out into the mist and off into the horizon and look to see whether or not these can provide benefit to patients in cancer. So we'll all find out together in 2026.
Talking to that, phase 1, phase 2, phase 3, a lot of biotech companies will maybe get through phase 1 and then they either license themselves or get sold or whatever if they start showing efficacious data or safety data, right? So you take the next steps. As you think about the development life cycle, obviously it takes a really long time to go through all three trials. How do you think about that? Do you see taking this thing all the way through? Do you see an acquisition prior? I know you can't totally speak to specifics there, but what are your thoughts on that?
So I think in this current, like, macroeconomic market, like, taking biotech even out of the picture, like, in this current macroeconomic market where, right, things are challenging, I think everything has to be on the table, regardless of what type of company you are, but, like, particularly even for biotech. So, like, the concept of, like, maybe you do a small partnership early and that's kind of like a try before you buy scenario, and they're like, well, we're going to test it out, and if we like you, we'll just buy you later, right? So, like, the M&A path, I think you've got to keep on the table, a classic IPO path, and then possibly you get acquired even after you IPO, or staying private and running the whole show yourself and going all the way and becoming a private revenue generating company, or being a public revenue generating company, or in licensing or out licensing different programs from other companies in order to bring in capital. So I think at this point, absolutely everything is on the table for Siren. We're not excluding anything. We're not gunning for any one of those. Everything is on the table in this market because I think you kind of have to. When it comes to how are we thinking about our clinical trials, the nice thing for us, right, folks may have heard of this, like, class of disorders called, like, rare diseases, and that it's not just like a buzzword. It actually has very important meanings when it comes to being a biotech company, because when you work in a rare disease, which is, right, we're only talking about the US for a moment, fewer than 200,000 patients in the US per year, then that rare disease is eligible for what's called orphan designation or orphan protections, which means that you're eligible for all these other FDA designations, fast track, breakthrough, accelerated, all these very fancy words that basically just mean there are regulatory speed ups that the FDA can offer you, one of the most important of which is that you can skip that phase 3 trial. And you can have your phase 2 trial be what's called pivotal and registrational. And then that's your last trial. And then you can go up for FDA approval. And then after your approval, you can run a phase 4 trial where now you actually kind of do what would have been considered a phase 3. But you can run it after you got an FDA approval. And it's in order to try to get things out to patients much more quickly so that these rare disease patients who often don't have options, often these are very fatal disorders, things like cancer, right? Where like you just don't have time to go through the classic 10 to 15 year pipeline to get a drug to market because you're going to die in the meantime. And so you can have these phase 1, 2 trials and skip a phase 3. And the nice thing for Siren, and one of the reasons that we're really prioritizing brain cancers, is that all 125 cancers that happen in the brain are all rare. And so that means all of our trials, in theory, right, we still have to apply for the designation, but in theory, they would all be eligible for these FDA designations that allow these speed ups so that we wouldn't need to run a phase 3 trial. And we actually just had a really productive and wonderful meeting with the FDA back in April, where we asked them to actually combine our phase 1 and our phase 2. Folks may have probably heard of a combined trial before, a combined phase 1/2 trial. So that way you only run one trial where there's no gap in time. As soon as you dose your last patient on your phase 1, you can then dose your first patient on your phase 2, and there's no gap in time. You don't have to reapply to the FDA. So you can combine those 2 trials and have a single pivotal and registrational combined phase 1 true, like all these buzzwords, apologies. But what that means is that you can really move much, much faster. And so it allows you to really kind of bring things to clinic much faster, get patients enrolled and dosed much faster, and hopefully bring it to the market much, much faster. And so that means we'll have clinical trial readouts much more quickly. That kind of brings all those possibilities you were mentioning on the table much earlier. So whether that's an M&A, whether it's an IPO, whether it's staying private but being a revenue generating company with an FDA approved drug, right? All of those become much, much sooner in the timeline, and they don't have to be like 2035, right? It could be like 2027 or 2028 or 2029, right? Much, much, much faster than would have historically been possible without working in a rare disease.
What is the phase 1 that you're developing right now going to look like? And I mean that in terms of like patient population, how you're going to be recruiting. Are there certain hospitals you're going to be working at? Like, what are some of the mechanics behind it and what it'll look like?
So the way this will be run, it's going to be a multi-site. We actually haven't decided internally yet, much less announced publicly what all the sites will be, but it'll be a multi-center trial, and that's on purpose. So that's one of the kind of FDA requests if you want to apply for those special designations. So things like fast track and accelerated and breakthrough. They want you to run multiple trials at multiple sites. They want that heterogeneity in the patient population and of the different treating physicians and of the different treating hospitals to really kind of get a better sense for what is the makeup of this very diverse population that you're going to have, rather than just having it at one hospital with one investigator at one site, right? They want that heterogeneity. And so it'll be a multi-center trial. And this is actually going to be in adults with recurrent high-grade gliomas. If that specific brain cancer doesn't mean anything to you in terms of what it is, it's just basically the gnarliest brain cancer there is. So it is one of the most serious and aggressive brain cancers that currently doesn't really have any effective drugs. And these patients, unfortunately, typically die within a year of diagnosis. And so pretty uniformly fatal cancer, really, really awful. And we really want to try to do something for these patients. And so we'll have a phase 1 here, hopefully in early 2026, so we can start enrolling patients as fast as we can.
So in terms of the animal studies that you've done, what data in there excites you that says, hey, we're going to be able to go in and kill off the gnarliest of cancers in the brain? Like, what did you see?
For sure. So there's kind of like 2 camps of data. There's what's called like basically just like normal data. That's like the normal data that everyone runs in, like things like mice and rats, where you have like genetic knockout mice or mice that you've given a particular disease that's going to test like the efficacy, right? Does it work? Yes, no. And then there's usually your large animal data, and that's where you test toxicity. So that's where you're looking for safety signals, right? And so we've now got both sets of these data. We've got large animal data, so we've actually done this in pigs. So pigs have a very similar brain anatomy to humans. It's also a very similar size and kind of shape and orientation and all of these things. And we had this beautiful study that we did last year in pigs with like the real drug, with the real like delivery device, with the real dose, with the real like flow rate, and neurosurgeons were there. We're doing live MRIs and like the whole setup, right? The real deal. And it was just like perfect. Like The pigs were fine. Everything was great. All the pathology after the fact looked great. So we were really excited from like a safety perspective because like safety is always first, right? Efficacy is second. Safety is always first. So we were super excited that for the first time being in large animals, right, there's always like a nervousness. Everyone starts in mice first because they're inexpensive and they're very simple models. You can do a lot of studies in mice. But when you switch up to large animals, things get really expensive very fast because, you know, right, like as you can imagine, right, like a pig takes up a whole lot more space than a mouse. So these are very, very, very expensive studies. And so to get into large animals for the first time and see that like it maintained all the safety signals that we wanted with no kind of adverse events was really exciting. And then on the efficacy side in mice, gosh, I think we're up to, you know, just shy of like 600 animals we've treated at this point where we've given them actual human brain tumors. So even though they're mice, we can give them human brain tumors. So you can take mice, these very special mice that have like a slightly modified immune system so that they won't reject human cells. And you can actually take from human patients, you can take brain tumors that were being resected anyways and that were going to go in the trash. You can take those human brain tumors and you can actually implant them into the mouse brain and they will grow into a mini human brain tumor inside these mice. So now you've got a living system, right, with blood flow and cerebrospinal fluid and all the things like happening like normal. It's not like a little tumor in a dish, right? It's a real growing brain tumor in a living animal. And then you can go and you can give, again, your human drug, not a mouse version of the drug, the human drug, because you're treating a human brain tumor in a living animal, and show in real time. We can do all this fancy imaging and we can watch these tumors grow and shrink in real time in response to different therapies, like all the standard of care drugs versus our drug called Siren 101 and all these types of things. And we're just seeing like remarkable, I mean, I'm like nervous, I'm like knock on wood. It just looks remarkable what we're able to see, where we see like 86% of the animals that we're treating are alive with complete responses, right? Like we can kind of use the capital C cure word, right? These mice no longer have the most aggressive brain cancer that there is, these recurrent high-grade gliomas. And so we're really excited about the potential. And so, yeah, between all of our small animal studies and our large animal studies spanning efficacy, biodistribution, and toxicity and all these things, just really, really excited. And so now we're actually running our very final study. This was the meeting we just had with the FDA last month, was to ask, like, what's the last study you want us to run before we do a human clinical trial? And they're like, we want you to do this one more study. And we actually just started dosing those pigs last week. So that study is ongoing and we'll read out this fall.
As investors, right, thinking about the market opportunity for something like this, obviously it sounds like a really big deal. Just in its first application, these kind of brain tumors, how big is the opportunity when you think about pricing? Like maybe give us some proxies of other, you know, specialized drugs that are out there. How big of an opportunity is this?
So it's one of those things, it's nuanced to model, right? Because if you compare it to other cancer drugs, right, the other cancer drugs in the brain all don't work. Right? We're comparing against things that don't work. It's like, how much does this car work that doesn't run? And you're like, nothing. Right? So if you then, right, so it's hard to price things in the brain because there are no effective drugs in the brain. There just aren't. There are no effective drugs for brain cancers. So we can compare to those prices, but it's like you're not comparing to anything meaningful. We can go into other cancer spaces like breast cancer, lung cancer, like other solid tumor spaces where there are effective drugs that do massively provide benefit to patients. And then you can kind of start to compare those numbers. But again, now it gets nuanced where it's like, well, if that provides 6 months of benefit, should that be priced the same as something that provides years of benefit? What if two drugs have the exact same number of months of benefit? You get 9 extra months, but one makes you more sick than the other one. Should that be priced the same? So pricing nuances are like, whew, that's like a 5 years from now problem. Also, like, what will the market tolerate, right? I think particularly in this particular economy and with this administration and all the changes that are happening at the FDA and all these types of things, I think the world 5 years from now, when like a new drug, whether it's us or someone else, comes to market, will be a completely different world, right? We're about to have, you know, a bunch of big changes at CMS, right? The group that runs Medicare and Medi-Cal and Medicaid, which is a vast majority of cancer patients actually get their insurance through these types of programs, right? Because cancer tends to skew towards older adults, folks who tend to be closer to retirement age. And so what is a price that will be covered by CMS 5 years from now? My goodness, I don't know. But no matter what, right, there's, even in just this first indication, we're looking at, this is tens of thousands of patients in just the US market per year. And so regardless of whether it's priced at $10,000, $100,000, $500,000, whatever it is, there's a very large patient population to go after. And so there's definitely a big market opportunity. The exact number, I would be lying through my teeth if I told you what it was, because no one knows what it's going to be, but there is a very large patient population, so it'll be a robust, healthy market.
One other question for you here. I think you mentioned there's something like 135 of these unique kind of cancer tumors that could be in the brain. What challenges does that present in terms of how you tackle each one? Is it wholly different, or is it pretty much the same thing with slight modifications? And then as you think more broadly, does this go to all cancers across the entire body, or does it really have to be to specific areas?
Yeah, absolutely. So some brain cancers are going to be like carbon copies of each other in terms of like how you would set up the clinical trial, how you would dose those patients, what the clinical trial kind of organization and setup would look like, what the time points will be, what the samples are that you're going to take, what the endpoints are going to be. Some of these are just going to be super cookie cutter. And those are obviously the ones that we would be interested in going after as like our second and third and fourth indications, these types of things, because it will be so easy and so fast to leverage. Because now not only is the tissue the same, right? It's the brain. But now like the delivery device is the same, the delivery route is the same, the drug, the actual drug is the same. It's not a unique drug that we have to redevelop every time. It's the exact same drug. It's just Siren 101, given at the same dose, with the same flow rate, with the same clinical trial design, probably at the same institutions, with the same PIs at each institution, with the same endpoints, and it just becomes very cookie cutter, which means right from a patient perspective, that's great because we can enroll much faster. These are much simpler trials. The cost to run the trial and these types of things will be much less, and so we don't have to queue up like, a $350 million Series E, right? Like you might be able to run a trial like from start to finish, right? From the beginning to the end. It might only be that you might need to raise, $100 million rather than, right? A normal drug to go from start to finish is often north of $2 billion with a B, $billion and 10 to 15 years. I think especially as we start incorporating all of these AI advances and these types of things, in terms of clinical trial recruitment and paperwork and IND filing with the FDA and all of the cool things that they're talking about implementing for rare diseases in terms of regulatory speedups, I think we could get to a world where we shorten that 10-year window down to single-digit years. And I'm not talking like 9, going from 10 to 9. I mean going from like 10 to 4 or something, like a big dramatic increase, and no longer needing to raise like $2 billion. It might only be that you need to raise maybe $100 million or $200 million for each new program. And so I think we are going to get to a time and a place in biotech and in medicine where these things will be coming online much, much faster, and it makes it so much easier to go after. And so we would definitely prioritize the indications that will allow us to do that kind of cookie cutter type process in the brain and just go from, right, high-grade gliomas to astrocytomas to oligodendriomas to medulloblastomas to meningiomas, right? There's 125 of them. And really prioritizing the ones that'll allow us to kind of move as rapidly as possible to get to those patients as quickly as possible.
For those investors who are listening, I'll give you the last word, who are kind of on the line, they haven't made their decision yet. What would you say? What do you want them to know?
The single best data point I can ever give anybody, whether you're an investor or not, on how excited I am about the data and how much I believe in the company is that I quit my job. Right? How many professors do you know that quit their job of being a professor to actually go start and run the company? Not sit on the scientific advisory board, not like be an acting interim CSO, quit their job, didn't take sabbatical, didn't take leave, right? Quit their job to go be the CEO. That's the best data point I can give anybody. This is the most exciting data I've seen in like 17 years in the space.
If you liked what you heard on the show today, you can check out why Siren Biotechnology received a 3.6 out of 5 and an investment from Kingscrowd Capital at kingscrowd.com. You can now invest on WeFunder until July 9th, 2025. And please like, subscribe, and share our podcast wherever you listen to podcasts so more listeners like you can find us. Thanks for listening.